Having just covered a couple of pre-processing steps for DTI data, namely distortion correction with top-up and eddy correction with the eddy tool, we're now ready to move on to actually creating and fitting the tensors. And this will create these FA or fractional anisotropy maps. So to do this will look pretty similar. We did it with some example data just a few videos ago. But all we do is go to the GUI and open up FDT diffusion. I'm doing this from the directory where I just did the eddy current correction and also top up. I select DTI fit and specify input my files manually because I just feel a little bit better doing that. We're going to select eddy unwarped images for the diffusion weighted data, everything that we just did all the pre-processing for, and also we selected a mask right here. Okay, so whatever you've generated using BET, the brain extraction tool. We will also supply the gradient directions in the BVEX file and BVALs as well. Click go, and this process only takes about 10 seconds or so. Once all the slices are processed, we can go ahead and look at those tensors just like we did previously. So that's done. Go to FSL view just to make sure that this looks correct. Again, typically you try to do this for every single subject just to make sure nothing went terribly, horribly, horrendously wrong. First we're going to open up the DTI_FA, which was just generated. And to look at these tensors, let's add the principal component, this V1 right here, DTI V1. Now remember, we click on this I down here and select the image type to be diffusion tensor. We can then display as RGB and modulate it by DTI FA. So what's going to be brightest showing up in this map is where the diffusion is the greatest in a particular direction. Okay, so that is just the fractional anisotropy step. We actually create the tensors, and then the rest of it is just going to be some more processing and then the actual statistical analysis to compare tracts between, let's say, two groups. Now that we've run our pre-processing and generated our FA, or fractional anisotropy maps, we're ready to do another acronym, which is called TBSS, or Tract-Based Spatial Statistics. Now, in this case, every command that we're going to use after we have these FA images is going to be prefixed with TBSS. So for example, TBSS_1 _preproc. And these things are all labeled pretty well in the FSL binaries directory. So let's take a look at what we're going to do. First of all, just to give you an overview, after we have all of our fractional anisotropy maps, we put them all in a single directory. And just for the record, I'm using the pre-cooked data, all the stuff online on the FSL website, and I'm in FDT1/TBSS. So these 5, there used to be 6, I kind of screwed up and erased one of them, and instead of just re-downloading it, I'm just going to kind of push forward. But we actually do have all 6 in these other pre-cooked directories, which have already been analyzed, the pre-computed registration and the stats. So just for the first step, it's not going to be exactly the same, but everything else after that will be fine. Just a little caveat, yeah, I'm kind of lazy. Didn't want to re-download it. But anyway, so imagine that these are all of the subjects in your study. All these .nai.gz files are the FA maps that have been processed like we just did in the previous couple of videos, and now they're all in a single directory. We're going to use these different TBSS tools, which fortunately enough are labeled in order to do all the pre-processing before we analyze it with the general linear model. So the first command, I said all these are prefixed by TBSS, is TBSS_1 _preproc. And all you need to do is give it all of the different FA files you have in your directory. You can either specify them individually or just grab them all with a wildcard like I've done right here. And while that's going, I would just like to show you where all this stuff is in the FSL directory binaries, subdirectory. So all these commands with this TBSS, those are pretty much all that's going to be called upon after we have our FA images and we're trying to prepare it for entry into the GLM. So the ones that I'm going to be using here are 1, 2, 3, and 4. Again, we run these sequentially. It's very helpfully labeled. Preproc is going to do some more erosion of the mask to get rid of any artifacts at the edge of those FA maps, and registration post-registration, just like in traditional fMRI analysis. You're going to estimate some warping parameters to a standardized space and then apply it with this post-reg command. And lastly, pre-stats is going to project all this pre-aligned FA data onto this track skeleton, which we'll see in just a second. So that's for your own edification. You can also open these things up on your own if you want, which can also be useful. So if you want to see what happens in step 4, you can just open it up with your favorite editor. Here I've used the VI editor, and you can see what it does, assuming that you know the bash shell and some basic Unix. Awesome. Okay. So the first preprocessing step is finished, and it recommends using slices dir, whatever's in there, to view what's just happened. And this is an important step in any step of neuroimaging analysis. After you do something, pretty much every time, I'm just going to lay this down as a rule. I don't always follow it, but it's a good thing to just keep in mind and to feel guilty about when you don't do it, is to look at your data after each step. Okay, so that's what we are going to do. Let's go into this. So it's generated an FA directory, which now contains all of our preprocessed FA images after that first step, and also slices dir right over here. Okay, so let's take a look at that. Let's open it up with Firefox, for example, and I think it's called index.html. So quickly open that up, and we get a good sense that it doesn't look like anything has gone horribly wrong. It seems like the erosion has been totally appropriate, and we're good to go on to the next step. You could also load these into FSL View if you wanted to take a more detailed analysis, but just for a quick run through, that is perfectly acceptable to my eyes. Okay, so now you would run the rest of these tbss commands from this directory. So one directory above where all that data has now been shoveled into this FA directory. So for example, tbss_2_reg, we would run from this directory. And notice, you can take a quick look at the help. It has just a few options you can select from. And most of the time, just use the recommended. So for example, I would just give this a -T, capital T flag, to use this template that they've created right here. Okay, so if I were to run that, I would just do this, hit enter, and off it would go. However, that takes a long time, and it's already been pre-computed, so we're just going to copy everything in this pre-computed registration folder and put it over into that FA folder. So just as if we had just run that step right now. So we just covered tools 1 and 2, some erosion of the images, and also some registration parameters for warping, which we will now cover with TBS tool number 3. And after that, we will then project all that onto a skeleton with TBSS4. So let's get right into it. Again, remember, I'm running all of these from this top-level directory. We're one directory above this FA directory containing everything that we've done so far. Okay, so if I were to run TBSS3, notice it only has 2 options. One is capital S, which you derive the mean FA and the mean FA skeleton, which is roughly where we're going to be doing the analysis of our DTI data. It's going to derive that from the mean of all subjects in the study. And capital T means use a template provided by FSL. Either one of these is fine. I mean, I always use the recommended one, but you can use whatever you want. Compare, just do whatever. I don't think it's that big of a deal. So if I did run that, again, this takes a long time, and I already have this all processed. But if I were to do that, we'd have this new directory right here called stats. So now we're starting to fill in things like, what is the mean FA skeleton? Okay, and it's also going to merge all of our different FA maps so that we can compare each of them against this skeleton that we've just created. So first what I'm going to do is I'm going to overlay this skeleton that we just made, or the mean FA map on top of a standardized template, just to make sure that the registration went okay and nothing went horribly, terribly, horrifically wrong. So I'm going to use the Enigmau That's a good template. That's the space that was registered to, so we should be able to see it there. And I'm going to use the color scheme of red minus yellow, which is just like a heat map. And the boundaries of the intensities 0.2 to 0.6 is pretty good. And so once we load that up, we're just going to compare this white matter mask on top of this template. Looks pretty good, seems to follow these white matter pathways pretty well. Doesn't seem to encroach too much on things like CSF or gray matter, so this looks pretty good so far. And the last thing that we're going to inspect from the output of the TBSS3 command is this merged image of all the FA maps. So we simply merged all the FA maps of all these different subjects in our study. Okay, here it's only 6 people, but you would do it for everybody in your own study. Okay, so it could be a lot. And again, we're going to use a slightly different color scheme, green, and same boundaries, 0.2 to 0.6. But the important thing is we'll be able to scroll through each one of these different subjects. So if I highlight all FA, okay, notice in green this is traced out the skeleton, right? And it's traced out a good representation of where these white matter tracks are. If I highlight all FA, notice volume 0 is the first subject in that. So as I click through it, I'm going to be seeing how does it overlay on top of each of my subjects, okay? And it seems to do a pretty good job. So we're just making sure that it's going to be a good candidate for doing a group level analysis. That it's actually going to be looking at white matter and not something else. That's too crazy. All right, so the final TBSS step before doing our GLM is TBSS4, naturally enough. And all this is going to do is basically it works the way any other kind of thresholding does, like thresholding for masks, thresholding for brain extraction. We're trying to just get a good sense and be as sensible as we can about where we're going to be doing our statistics. So this is going to further threshold that skeleton, and it's also going to be doing some distance estimation of the skeleton. So not to get into too much detail, but basically it's just going to make sure that there's not going to be any strange connections between different parts of the white matter which should not connect. Okay. So let's say I ran that and I used the recommended threshold of 0.2. Again, this stuff gets sent back into the stats directory, and we're going to take a very brief look at it. Okay, so the output from this is going to be this all FA skeletonized data set. And again, once we have that, we're going to look through each subject just to make sure that nothing looks too out of whack, that there's relatively good alignment, and that the values seem to be sensible, that there's nothing too extreme here. So, we could highlight one voxel within this track right here. Say, it has an intensity of 0.7. And as we go through each of these different subjects, just make sure, there's nobody that's a huge outlier. Obviously, there's going to be some variability. But in general, just make sure this looks fine. You can turn on movie mode if you want. Actually, let's slow it down a little. Just chill with this, okay? So I selected options. We're going to go to the MISC tab, and let's make the frame rate 500 milliseconds. So if we turn that on again, this is just another way that you can look at each subject. If you have a lot of people, that beats just clicking on each of that individually. So anyway, those are the TBSS tools, and now we're ready to do randomize and also creating a design matrix and a contrast vector, which we can use to compare these two different groups that we have and analyze any differences in their tractography. That's what we'll get to in the next tutorial. But these first 4 steps should be pretty straightforward, especially since they're labeled very easily, very sensibly. Just in increasing order, you do 1, then 2, then 3, then 4. It really generates the entire directory structure for you, and as long as you just look at the data after each step and make sure nothing went too out of whack, this will put everything in a good place for doing your actual analysis. Okay, so hope that helps. I will see you guys in the next tutorial, and we'll finish up this sample data set with FSL.